Hello. So today we are going to see the topic of cell. Cell will comes under the third unit of class 11 NCERT biology. So the unit name will be cell, structure as well as function. Under this unit we have three chapters. First one is cell, the basic unit of life, and the next one is cell cycle and cell division, and the third one is biomolecules. So let's see about the cell. What and all the important things we have to be learned regarding the topic of cell. Coming to the cell, so cell is considered as the basic structural as well as fundamental unit of life. So the fundamental basic structural as well as functional unit of life is cell. So to this entire statement, so far before much more work has been done. So what is the work? So the work is nothing but it is bring upon many cell theory, many scientists. So the very first scientist we remember when it comes to the cell is Robert Hooke. Robert Hooke first observed under the microscope the cork cells. So we all know the cork cells are actually dead cells. There is no cytoplasm as well as protoplasm in that. So what he observed under the microscope is a dead cell wall. Okay, so only the cell wall which is non-living material. So the non-living cell wall material he observed under the microscope and he named that open spaces as cell. Okay, cell is nothing but open chambers which doesn't consist of anything in that. And he has first given the term cell in his book called Micrographia. Okay, so now let's see the other scientist's name. So who has given the cell theory? So the cell is considered as the basic unit of life. The cell is considered as the structural unit of life. The cell is considered as the functional unit of life. These things are actually given from the cell theory. So what the cell theory states? The cell theory is actually proposed by Sheldon and Swan. Sheldon is a botanist and Swan is a zoologist. They both has been put forward the cell theory and finally has been confined that all the plant is being made up of group of different types of cells. All the animals are being made up of different kinds of cells. So entire the plants and animals are being made up of all different types of cells. So this is confined from the cell theory which is given by Sheldon and Swan. Next, who have been first observed and identified the living cells? So the living cell was first identified, the father of microbiology. The father of microbiology is none other than Anton von Leeuwenhoek. So what Anton von Leeuwenhoek what has observed is he is actually a lens maker. Under his own homemade microscope, he observed the bacteria, he observed the RBC, he observed the sperms, he observed the tooth scrapings, he observed the rainwater. In all these things, he found very small things. And also he named that, so he has seen many different movement things under the microscope. So first living cell is being observed by Anton von Leeuwenhoek. So they both put forward the cell theory, but one point that is been lacking with cell theory. So what is lacking with cell theory is where the cells came actually. So the cells came actually, they couldn't able to answer from the cell theory how the cells got originated. Later, the scientist called Rudolf Virchow. So Rudolf Virchow has been put forward the point all the cells came from pre-existing cells. From the Latinized statement that omnis cellula e cellula. So the meaning of omnis cellula e cellula is all the cells came from pre-existing cells. Got it? So this we call it as omnis cellula e cellula. This is very important previous question from NEET as well as All India Medical Entrance Test. So in many of the examinations. And he is the one who completed the cell theory actually. And next. So what are the different types of cells we have? So we have to remember certain scientist name. One is Robert Hooke and other one is Anton von Leeuwenhoek. And next Sheldon and Swan and Rudolf Virchow and Singer and Nicholson. Singer and Nicholson gave the fluid mosaic model. So far we are going to see different types of cells. How so far the cells? So we have majorly based on the cell type and based on the nuclear membrane, we have prokaryotic as well as eukaryotic cell. Okay, and next we have the plant cell as well as animal cell. Next, unicellular as well as multicellular. So these are the different types of cells we have. And coming to the variety of cells, that is, which is the largest cell so far on the entire earth and the entire biosphere. So that is ostrich egg is being identified as the largest cell among all the living things. And which is the smallest cell? Smallest cell is nothing but Mycoplasma gallisepticum, which is measured around 0.1 micrometers. So that is been identified as the smallest cell in the entire biosphere. The largest cell is said to be the ostrich egg. Okay, and next let's see about what is prokaryotic and what is eukaryotic. Coming to the prokaryotic, the kingdom Monera, all the living organisms that comes under the kingdom Monera, we call it as prokaryotic. What does the prokaryotic consist of? The prokaryotic doesn't have any nuclear membrane. Okay, and the prokaryotic is the thing which has no cell or membrane bounded cell organelles will be absent. So starting from outside to inside, so in under prokaryotes you will be having cell wall. So the very outer layer we call it as capsule or slime layer. The layer, if it is very tough and rigid, then we call it as the capsule. If the layer is loosely arranged, then we call it as slime layer. And later we have the cell wall. The cell wall is made up of peptidoglycan. So, and the peptidoglycan consists of N-acetyl glucosamine and N-acetyl muramic acid. So that's why the material we call it as murin. And next we have cell membrane. Cell membrane will be very similar to eukaryotic cell membrane. So phospholipid bilayer, proteins all together will be there. And the cell membrane is been having infoldings towards the cytoplasm, which consists of respiratory enzymes. This respiratory enzymes is for the respiration. You just remember the letter M. Mesosomes is for respiration for prokaryotic and mitochondria is for the respiration of eukaryotic. So that we got finished. And when it coming to the cytoplasm, we have inside the cytoplasm we have the nucleoid. Nucleoid is nothing but naked double-stranded DNA, which is like a loop. And the number of chromosomes for the prokaryotic is only one chromosome. And that too no histone proteins will be present. And coming to the plasmid. Plasmid is considered to be the extra chromosomal DNA. So that is also double-stranded. Actually, this extra chromosomal DNA, because of this only, the antibiotic resistance will be provided by the bacteria. It has a specific type of phenotypical character will be exhibited because of the plasmid. And next, in the cytoplasm, we have 70S ribosomes. The 70S ribosomes is being made up of 50S as well as 30S. Okay, and next coming to the chromatophores. We have various colored particles as well as the chlorophyll. We don't have a chloroplast. So when it comes to blue-green algae, okay, so when it coming to blue-green algae, we don't have the thing called chloroplast. We have the thing called rounded bodies, nothing but chlorophyll pigment will be present in that. And next we have cyanophyllin granules, glycogen droplets. So these things will be like floating bodies in the cytoplasm. And next to that, we have an outgrowth structures like flagella. Flagella is made up of a protein called flagellin, which doesn't have 9+2 arrangement. The 9+2 arrangement will not be there for flagella of the prokaryotic. And next, so coming to the next thing, we have pili as well as fimbriae. Pili is used for sexual reproduction. So we call it as sex pili as well. And fimbriae is used for the attachment of the bacteria on the rocks or on the hard surfaces. So these are the certain very important things of prokaryotes we have to remember. And all the bacteria are prokaryotes and all the monerans are prokaryotes. Remember one statement, all bacteria will comes under kingdom Monera, but not all monerans are bacteria. So the prokaryotes, various prokaryotics are bacteria, blue-green algae, Okay, so bacteria, blue-green algae, mycoplasma. So these things has to be blue-green algae, mycoplasma and actinomycetes. These are all considered as the prokaryotes. And next, coming to eukaryotes. Coming to the eukaryotes, so remaining all the kingdoms, starting from kingdom Protista to kingdom Animalia, all together we call it as eukaryotes. Okay, so coming to the eukaryotic, starting from outside to inside. So cell wall will be present for Protista, for fungi as well as for kingdom Plantae. For Animalia, the cell wall will not be there. So when it comes to the Protista, the cell wall is made up of calcium and silicon. And when you go to the fungi, the cell wall is made up of chitin. When you go with the plant kingdom, the cell wall is made up of cellulose, hemicellulose, pectin and lignin. So coming to after the cell wall, we have the cell membrane. Cell membrane is made up of phospholipid bilayer. So and we otherwise call it as fluid mosaic model, which is widely accepted model. So that is given by Singer and Nicholson. So 30% will be occupied by the proteins and remaining 70% of the thing will be occupied by the phospholipids. And in the phospholipids, we have head as well as the tail portion. Head will be the very exterior and tail portions will be interior. The head of the phospholipids are hydrophilic. The tail of the phospholipids are hydrophobic. That's why whatever the contents which are water soluble and proteins, which will be present very exteriorly. It will stick only exteriorly. It can't pass through the membrane. And if it is lipid and steroid made, it can pass through that cell membrane very easily. Okay. And next thing. So once you enter into the cell, we have certain membrane bounded cell organelles as well as non-membrane bounded cell organelles. Coming to the first thing, membrane bounded cell organelles. Under membrane bounded cell organelles, we have single membrane bounded cell organelles as well as double membrane bounded cell organelles. First, coming to single membrane bounded cell organelles, we have endomembrane system. So that is starting with endoplasmic reticulum. Endoplasmic reticulum is being first discovered by Porter, and actually it will be like a net. So that's why we call it as endoplasmic reticulum. And this endoplasmic reticulum is mainly for the synthesis of proteins as well as for the synthesis of lipids. And we consist of two types of endoplasmic reticulum. One is smooth endoplasmic reticulum and other one is rough endoplasmic reticulum. Smooth endoplasmic reticulum is mainly meant for lipid synthesis. Rough endoplasmic reticulum is mainly meant for protein synthesis. Okay, so you can find the smooth endoplasmic reticulum more in steroid hormone producing glands and rough endoplasmic reticulum in protein or polypeptide producing glands. And next we have the Golgi apparatus. Golgi apparatus has been first discovered by Camillo Golgi. That's why the name Golgi apparatus. That is actually for processing of the proteins as well as the lipids. So packing is actually done in the Golgi apparatus. So we have a marker enzymes in the Golgi apparatus, that is glycosyl transferases. This is so far very, very important, and marker enzymes. And next, coming to the part of vacuoles. The vacuoles are nothing but the warehouses of the cell. And the liquid which is present inside the vacuoles, we call it as cell sap. And the membrane, we call it as tonoplast. And next, coming to the next other thing, we have the lysosomes. We otherwise call atomic bombs of the cell or suicidal bags of the cell. Okay, so lysosome consists of lytic enzymes like amylolytic, proteolytic, so lipolytic enzymes will be there. And next, so these will comes under endomembrane system. So under endomembrane system, we have endoplasmic reticulum, Golgi apparatus, vacuole, and as well as lysosomes. These are single membrane bounded organelles, and we have the microsomes also, means minor rounded bodies, nothing but peroxisomes, which consists of hydrogen peroxide, glyoxysomes, spirosomes. Okay, so peroxisomes, glyoxysomes, spirosomes, microsomes, these are all also single membrane bounded cell organelles. These are minor cell organelles which will be floating in the cytoplasm. And next, we'll go with the double membrane bounded cell organelles. We have chloroplast, which is a double membrane bounded, mitochondria, and next nucleus. So chloroplast is meant for the photosynthesis. and mitochondria is meant for the powerhouse of the cell for the production of ATP. Okay, so next coming to the nucleus. Nucleus is considered as the brain of the cell. It has double membrane bounded. On the outer membrane we have the ribosomes will be stick on the outer membrane, and the membrane to membrane we have a pores. We call that as nucleopore, and inside the nucleus we have nucleolus, nucleoplasm, chromatin material, and the chromatin material is actually made up of histone proteins, non-histone proteins, as well as the DNA. Remember, the DNA is negatively charged and the histone proteins are positively charged. The histone proteins are mainly for packing the DNA with the negatively charged and positively charged histone proteins. Okay, and the next thing we are going to see is non-membrane bounded cell organelles, that is centrosomes, which is mainly helpful for spindle formation, that is only present in animal cell. So in plant cell you can't see the centrioles or the centrosomes. And next, ribosomes. Ribosomes are mainly called as protein factories. All the ribosomes align together during the process of translation. We call it as polysomes. And next, we have centrosomes and chromosomes also will not have any sort of membrane. So these we call it as no membrane bounded cell organelles. So this is about the prokaryotic as well as eukaryotic differentiation. Next, coming to the plant cell and animal cell differentiation. Plant cell consists of cell wall. Animal cell will not have any sort of cell wall. Plant cell consists of chloroplast. Animal cell will not have the chloroplast. Plant cell will not have the centriole or centrosome. So there is no spindle formation will be there, but not with centrosomes or centrioles during cell division. And next, when it comes to the animal kingdom, animal cell, it has the centriole or centrosomes, which leads to the formation of spindle fiber. And that is about key differences between the plant cell as well as the animal cell. And coming to the vacuole, one more thing. So large central vacuole will be present for the plant cell, and central vacuoles will not be there. If is there also, very minute will be there for animal cell. That is the main key differences between plant cell as well as animal cell. Next, unicellular and multicellular. Coming to the unicellular thing, so unicellular organisms will comes under kingdom Monera and kingdom Protista. So, and remaining kingdom fungi, kingdom plantae, kingdom animalia are multicellular organisms. Okay, that is the main differentiation between the unicellular as well as multicellular. And next, so these are all things will be studied under the topic of cell, the unit of life. Okay, so far we have seen the many important points from the previous year MCQs, and also so far exam expected topics and points. I hope this video will be very helpful to you. So please do subscribe to my channel for more very important updates. And so please make sure, take a note during this video, which will be very helpful for you to note down the important points for NEET preparation. So I hope you had a great informative video. Thank you very much.